This is James Leo. I'm doing lesson 11 group problems. Question 1 asks us, 1A asks us, indicate the group of enzymes that covalently link amino acids to tRNA molecules. In that case, that will be amino iso-tRNA synthase. Question 1B asks us to indicate 4 proteins that consume GTP during translation. That would be IF2, IF3, EF-TU, EF-G. 1C asks us to indicate 1 molecule that functions as a ribozyme during translation. That will be peptidotransferase. 1D asks us to indicate 2 proteins that recognize stop codons. That will be release factor, which includes RF1 and RF2. And finally, 1E asks us to indicate 1 molecule that forms hydrogen bonds with the Shin-Delgarno sequence in the mRNA. That will be the 16S rRNA. And in question number 2, it asks us to fill in the blanks. A, the first amino acid in prokaryotic protein is N-terminus. B, an E. coli protein called EF-TU delivers charged tRNAs to the A site of the ribosome during translation elongation. C, the initiator tRNA binds to the P site of the ribosome, whereas the remaining charged tRNAs bind to the A site of the ribosome. And D, release factor is the translation factor that helps the ribosome move 1 codon in the 3' direction along the mRNA. E, IF2 is a translation factor that guides the mRNA to the 30S ribosome subunit. F, AUG and UGA. So AUG codon is the start codon, while UGA is an example of the stop codon. G, degenerate, is a term that indicates that more than 1 codon can specify the same amino acid. H, the final amino acid is located at the C terminus of the polypeptide.
Hey guys, here we have week 11 group problems. Question 3 says a series of mRNA codons are indicated below. Consider the wobble rules and provide all possible tRNA anticodon sequences that can form hydrogen bonds with each mRNA codon. So I took a screenshot of the video that she had us watch. It was actually really helpful. And just remember that A can pair with U, C with G, just like normal. And G can pair with C or U with the wobble rules. U can pair with A or G. And the I, the alternate, can pair with A, C, and uracil. So with those rules, these are the answers I got. And question 4. Suppose that E. coli contains a protein called protein X, which is 218 kilodaltons in molecular weight. If the average amino acid is 125 daltons, how many amino acids are found in the protein? So you take the weight in daltons and you divide that by 1225, and you get 1744. That's how many amino acids there are. So B, if you know the number of amino acids, what is the length of the mRNA coding region? You can just multiply that by 3 to get the number of base pairs, which is 5,232. And we know that each base pair is 0.3 nanometers. So you get 1569.6. And C, how many GTP molecules are consumed to synthesize each copy of protein X? So there's 2 per amino acid, and then there's 1 to stop it at the end. So 2 times 1744 plus 1, you get 3,489. And then D, how many ATP molecules are consumed to synthesize each copy of protein X? That one's exactly the same as the amino acids, pretty easy. And what is the name of the group of enzymes that consumes ATP to assist in the synthesis of protein X? And I got aminoacyl tRNA synthase. Thanks, guys.
Hey, it's Dylan. I had problem 5. And so basically it was just talking about, it had A through E, and it gave us a list of different molecules, and it wanted us to decide which one was involved in each of the processes. I messed up on a couple, but after checking it over, this is what I have. So for A, it's which is involved with as a translation initiation. So that's going to be the 16S rRNA, the Shine-Delgarno sequence, the IF3, AUG, the start codon, tRNA, FMET, and then IF2. Second one is translation elongation. So that's also going to be the 16S rRNA. You're also going to have the EFTU, EFG, and the 23S rRNA. Now for C, it's translation termination. So we went through initiation and elongation, now we're on termination. So that's going to be the release factor 1, RF1, also the stop codon, UGA, and that's it. The next one is which of the following are parts of the 70S ribosome. So that's going to be the 16S rRNA and the 23S rRNA. We know that makes up parts of the 70S. And then E, the mRNA, which is found within mRNA, and the Shine-Delgarno sequence, the UGA, stop codon, sorry, the AUG, another codon, start codon, and I thought that this was, but it's not, because I realized that is an amino acid that attaches. So that's problem 5.